one of the things we do have to touch on in evolutionary biology is we have to look at how to make a cell. Now, the interesting thing about the theory of evolution and most of evolutionary biology is that we take for granted that cells exist. There's not a lot of people who are really looking at or trying to make a cell. We pick up when the cell has been made and then go from there. There is a small subset of scientists, mostly with biochemistry and cellular biology degrees, and what they're trying to do is they're trying to make the first synthetic cell, the first man-made or human-made life on this planet, and they're getting really close. So they're using carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. Those 6 atoms are elements that we find in all living things. They're making proteins, lipids, carbohydrates, and nucleic acids, and essentially they're trying to make the first unit of life that has been completely manufactured by humans. So cell theory states that the cell is the smallest unit of life and that cells come from pre-existing cells. So we're just briefly going to look at what it takes to make a cell and then go from there. So when it comes to making a cell, we know that we have those individual atoms that are bonding together, and that our atoms bond together to make molecules, and molecules come together, they start interacting with one another, and eventually we get the cell. Now, it's a huge leap to go from molecules interacting with one another to a cell, the smallest unit of life, controlling its interaction with the environment, controlling its internal environment, maintaining those systems within thresholds, in other words, maintaining homeostasis, using and obtaining energy, making more of itself, making genetically new combinations. We know that a cell can demonstrate all of those characteristics of life, but molecules cannot. So let's take a look quickly at some of the things that are necessary to make a cell. Now, when we talk about a cell has to have some kind of boundary, defining the inside and defining the outside. And this is why a plasma membrane becomes so crucial, why we focus in on it, why we go into detail with the parts of a plasma membrane and how those parts come together, and then how they're going to define essentially our smallest unit of life. So when we're looking at a plasma membrane is going to be made of phospholipids. And our phospholipids are going to have a bunch of atoms bonded together to make molecules to make something called the hydrophilic head. Now, the word hydrophilic means water-loving, so this part of our molecule is going to be attracted to water. We're going to have a fatty acid chain. That fatty acid chain is going to be carbon atoms and hydrogen atoms bonded together. Now, this fatty acid chain is going to be hydrophobic. The term hydrophobic means water repelling. So already when we look at this phospholipid molecule, we see a lot of molecular tension. We've got part of the molecule that's like, ooh, yes, I want to get at the water. And the other part of the molecule that's going, oh, ick, no, I hate water. I'm repelling the water. We've got this hydrophilic, hydrophobic situation. So within each individual molecule, we've got a lot of tension. That's going to be a really key point. Now, right here, I've just drawn a phospholipid that has just one tail. And one of the cool things about phospholipids is that when you get these one-tailed phospholipids all together in a jar of water and you shake them up, what's going to happen is these phospholipids are going to start to glob together in these little balls. Now, I can only draw this two-dimensionally because of the limitations right now with technology, but this glob of one-tailed phospholipids, where all of those hydrophilic heads are going to be in this three-dimensional kind of blobby shape. All those hydrophilic heads are going to be on the outside, loving and reacting with the water. And all of those tails are going to be on the inside, repelling and moving away from the water. So every single time you get a bunch of these one-tailed phospholipids into a beaker of water and give them a good shake, they're going to form this mycel, this glob. Now, the problem with this glob is that we don't have a defined inside. We've got a way to keep the outside out, right? To keep all that water out of our glob, but we don't have a defined inside. And the key thing with the cell is you've got to have a defined inside. You've got to have stuff trapped on the inside of this structure, stuff that's going to be interacting and being able to have reactions and being able to do all kinds of fun things. So the one-tailed phospholipid, while necessary in chemistry and really fun to play around with, is not going to get us closer to making the first cell, making that smallest unit of life. So this is why we focus in so much on the two-tailed phospholipids. So we still have all of our atoms bonded together to make our molecules of the hydrophilic head. And with our two-tailed phospholipid, we're going to have two fatty acid chains. They're still going to be hydrophobic. One tail is going to be called a saturated fatty acid chain. These are carbon atoms that have bonded to as many hydrogen atoms as possible. So every carbon atom is full of hydrogen, and it forces that fatty acid chain, that tail, to be straight and stable. Our other tail, our other fatty acid chain, is going to have a double bond between two of the carbon atoms. And that is happening right here in my picture. This double bond between those carbon atoms, where the two carbons have bonded to each other instead of bonding to hydrogen, means that not all of our carbon atoms are full of hydrogen. So this fatty acid chain is called unsaturated, and this unsaturated fatty acid chain has that very characteristic kink. That kink makes it a little bit more fluid, makes it a little bit less stable. So we have more molecular tension in this two-tailed phospholipid. We still have the hydrophilic, hydrophobic molecular tension. But then within the fatty acid chains, within these two tails, we see one tail being straight and saturated and the other tail being unsaturated and kind of fluid and kind of loosey-goosey. So a lot of molecular tension. If you get a bunch of these two-tailed phospholipids into a jar of water and give them a good shake, what's going to happen is these hydrophilic heads are going to sit close to each other. They never really quite touch. I find that a little bit creepy, especially considering the fact that we need this to be a very stable, solid boundary. Well, the solid is possibly I'll get into that in just a minute. So all of our hydrophilic heads will sit very close to one another. And there are thousands of different types of phospholipids. So that's why I'm drawing some of my unsaturated fatty acid chains in other directions. So they'll form kind of a single layer. And then the hydrophobic tails will bond to each other and will form this second layer. Now again, this should be a three-dimensional structure coming out in space as all of these phospholipids start to assemble. We get this bilayer of phospholipids, two molecules thick, two phospholipids thick. And all of our cell membranes, a lot of our plasma membranes will be this bilayer of two phospholipids. All of our hydrophilic heads will be interacting with water outside. All of our hydrophilic heads will be interacting with the water and liquid inside. In between our phospholipids, there can be no water because we've got this thick mass of hydrophobic tails. That is what defines a cell. We've got an outside, we've got an inside. We've got a way to control our interaction with the environment because of this mass of hydrophobic tails. Unfortunately, just because we've got this nice tight little layer, that doesn't necessarily mean that we can be a living entity. We need to be able to get things out, like wastes, get things in, like resources, so we have to have interaction with the environment. And this is where we're going to see embedded in this phospholipid bilayer all kinds of proteins, all kinds of carbohydrates, There will be some cholesterol molecules, and those cholesterol molecules will keep all of these phospholipids steady and stable, give them some kind of stability, because remember, there's a lot of molecular tension. Some of the creepy molecular tension that occurs is the idea that a phospholipid that previously had been interacting with the outside would be like, yeah, I'm sick of this job. I'm going to flip on into the inside and see what's going on. And this one that's been interacting on the inside could flip and interact, move itself to interact with the outside. So our phospholipid is constantly in motion. That should make us all feel a little creepy, because when we look down at our cells, we don't expect for our cells to be constantly moving. We like to think that this is a really, really, really tight barrier, and that's not constantly interacting with the environment, and that everything is nice and stable. Feels like it's stable. But at the molecular level, everything is wiggling and jiggling, and it's not completely solid. So some of the weird things that can happen, gases like oxygen can just slip between these phospholipids. Gases like carbon dioxide can just slip between. They don't even need a protein helper. They just get to come and go however they please. Things like water, though, they're going to need a protein helper. Remember all those hydrophobic tails. Water's not going to be able to cross this barrier unless it's got a helper. So plasma membranes, especially the cell membrane, that outer layer defining a cell, is absolutely the most important structure when we're looking at defining a cell, making a cell, having the smallest unit of life. So now that we've talked a little bit about the plasma membrane, there are a couple of key things that scientists have been doing to try and make a cell. The first thing that scientists did was they got phospholipids, two-tailed phospholipids like this, put them in a container, shook them up. They also had carbohydrates, lipids, and nucleic acids, and proteins. They had a whole lot of those other macromolecules. When they gave it a big shake, they found that the stuff on the inside started to interact. That was cool. They even started to see some control and interaction in and out across the plasma membrane. Well, that looks a lot like a cell. The protocells that they were making, though, had some big flaws. First of all, they didn't have little compartments on the inside. There seemed to be kind of a free-for-all happening as far as the molecular reactions inside of their protocell. So scientists kept working. Another structure that's been made a lot in biochemistry are these things called coacervates. These are similar kinds of structures. They have a separation of inside and outside. They can make more of themselves. But again, we don't have that compartmentalization. It wasn't until scientists started making microspheres that we started to see something that was closer to a cell. With the microsphere, we've got that ability to make compartments on the inside. Now, the reason why I'm emphasizing this and why we start to talk about the compartmentalization inside of our cell, after we've already made our cell membrane, is because of the different cell types. Now, there's two main cell types on this planet. One cell type is a prokaryotic cell, and a prokaryotic cell doesn't have these little compartments. A prokaryotic cell does not have a nucleus, and a nucleus is a plasma membrane sac of DNA, a compartment of DNA. A prokaryotic cell does not have all of these membrane-bound organelles. In other words, they don't have these plasma membrane organelles separating the biochemistry. They don't have things like mitochondria, Golgi apparatus, lysosome. All of the chemical reactions are just happening inside of the cell. A little bit like what we see with protocells and coacervates. So, a prokaryotic cell can make more of itself. It can use and obtain, and use energy. It can make genetically new combinations. It can demonstrate all of the characteristics of life. Prokaryotic cells are what we think of when we think of the first life on this planet. It was the first attempt at living things. Now, all of the species in the domain bacteria and all of the species in the domain archaea are made of these prokaryotic cells. So when we're studying the species of bacteria and we're studying the species of archaea, one of the questions that's always in the back of the minds of the scientists is, how are these species, demonstrating some of the first attempts of life on this planet. Now, eukaryotic cells are a lot more complicated. With eukaryotic cells, we have a plasma membrane-bound nucleus. We have a compartment for the DNA. We have plasma membrane-bound organelles, things like chloroplasts and mitochondria and Golgi apparatuses and lysosomes and rough and smooth endoplasmic reticulums. We have all of those compartments for biochemistry. A eukaryotic cell is controlling its interactions with all of these organelles and all of these compartments. So eukaryotic cells in evolution, we think came after prokaryotic cells. With eukaryotic cells, all of the species in the domain eukarya have these eukaryotic cells, and eukaryotic cells have helped some species to become multicellular. Now, when we think about multicellularity, this is a very strange concept. The idea that all of these individual cells that could live on their own have given up the ability to live on their own, they've specialized, and they formed an agreement with other cells. Some of the cells are going to do protection. Some of the cells are going to be obtaining energy. Other cells are going to be in charge of reproduction and making genetically new combinations. One cell doesn't have to do it all. We most of the time focus on multicellularity. We take it for granted, but it's very, very strange. We walk around as this whole organism. We feel like all of these cells are under control, like they're all part of us. But the one thing that we're taking for granted is the idea that all of our cells have a shared mission in being part of this organism, being part of the tissues that become part of the organs, part of the organ system, part of the entire organism. It isn't always the case. When we look at cancerous cells, one of the identifiers of a cancerous cell is that that cell has lost its identity as part of the tissue, as part of the organ. It's no longer part of the whole. And that's disturbing. That's a biological betrayal, because all of these cells are supposed to be working together. We don't want a few cells becoming weird and splitting off and going, nah, I don't want to be part of the lung. I'm not going to do that anymore. I'm going to go be lung cancer. Whoa. We're still trying to figure all of this out. But please remember, as we progress through this semester, we have to always take a step back and go, what kind of assumptions do we have when we're observing the system? Could those assumptions be influencing our conclusion? And right at the level of talking about cells, we know that we have a lot of assumptions, and those assumptions get in our way of being able to even see and observe what the system is doing, much less being able to understand what is going on. So, we end this chapter with a couple of main concepts focusing on how cells make more of themselves, touching base with those characteristics of life. If we have a prokaryotic cell can make more of itself by doing binary fission. Binary fission can be very quick. Of course it can. You don't have all those organelles inside to have to make copies of. So, for some species that have prokaryotic cells, one cell can make two genetically identical cells as quickly as 20 minutes. When we look at eukaryotic cells are going to have to do mitosis. And if you've been through a cell biology class, you know that mitosis involves interphase. G1, S, G2, then we get into the mitotic M phases, prophase, metaphase, anaphase, telophase. One cell will make two genetically identical cells, but it's going to take about 24 hours for this to happen, and it's way more complicated. Don't worry, in evolutionary biology, we're not going to get into all those details. But we do want to understand that both prokaryotic and eukaryotic cells can make exact copies. They can demonstrate the characteristic of growth. For prokaryotic cells, it's binary fission because the mechanism and the mechanics are different than eukaryotic cells, where the cell division process is mitosis. Both cell types can demonstrate that characteristic of life, reproduction, making genetically new combinations. So what a prokaryotic cell, like a bacterial cell, will do is it will start to slither up next to another prokaryotic cell. it will start to push out its plasma membrane. It will make a connection with that other prokaryotic cell, and then it'll start to swap genes back and forth. Molecularly, what that cell is asking is, hey man, you want some genes? I got some genes. Let me give you some genes. And so we see that gene swapping back and forth. This is called horizontal gene transfer, and it's done through a structure called the conjunction pili. That's what my arm reaching out is. Now for eukaryotic cells to make genetically new combinations, they're going to go through a much more complicated process. Again, we take this for granted because it's the way how we do reproduction as a human species. So in order for a lot of eukaryotic celled organisms to swap genes and make genetically new combinations, they're first going to make two separate organisms. One organism will make only gametes, like the egg. A separate organism will make only gametes like the sperm. So we have these two separate specialized cells, sperm and egg, and separate organisms are making the sperm and the egg. Just because you make these gamete cells, sperm and egg, that doesn't mean that you've made a genetically new combination. You've now got to get these gamete cells together. Now you're going to need to grow big antlers. You're going to need to have beautiful feathers, sing beautiful songs. You're going to have wine and chocolate and comfortable beds, and nobody better have a headache in order to get the sperm and the egg to come together in order for fertilization to happen to make the diploid zygote, the baby, the offspring, the next generation. That kid, baby, offspring, diploid zygote, that organism will have the genetically new combination. And when you think about it, all of this specialization, having to get the cells to come together in fertilization, and then that offspring growing, that is a lot of work. I mean, it would be so much easier if you could just slither up next to another organism, push out a little conjunction pili like a prokaryotic cell, swap some genes, and then you're on your way, and you've made your genetically new combination. But no. A lot of eukaryotic celled organisms are going to do it in a much more complicated way. So this is our brief overview of the origin of cells and a couple of those characteristics of life that are so crucial when we're looking for whether or not something is a living organism. Now we do end this chapter by talking about some things that are not living. One of those things that's not living is a prion. It's a misfolded protein, but it almost looks like it demonstrates some of the characteristics of life. And then we've got this this thing called a virus. And a virus is a protein capsule with some information inside. That information could be a DNA molecule, it could be an RNA molecule. And the weird thing about viruses is that viruses can accumulate mutations. So that looks a lot like genetically new combinations. Looks a lot like that characteristic of life reproduction. The virus can persist for a long period of time. Viruses they can go into what looks almost like a dormant state for a long period of time and then still infect living organisms later. Viruses can stick their DNA or RNA into living cells. And that's the really crucial thing here, because a virus can't make more of itself. A virus can't just make copies of its information, DNA or RNA, and then get bigger and pop off a whole new virus. No. The virus has to stick its information inside of the cell. The cell then reads the DNA or the RNA and then makes more viruses. The virus co-opts the cell into making more of itself. And that's really evil. That's really, really kind of mean. There is a part of biology that is looking at whether viruses might have been the first attempt at cellular life. Whoa! That gets really spooky really quickly. If you have more questions, if you start to get curious about all of this, please, by all means, send me emails. But it does get really complicated really quickly. We need to stay rooted in the idea that the cell is the smallest unit of life. Cells can make more of themselves. We have prokaryotic and eukaryotic cells, and the only way we're going to have those is with the phospholipids, the two-tailed phospholipids, making that plasma membrane, that bilayer. And that right now, viruses are considered non-living entities that can have a huge effect on living entities. If you get questions, please email me or drop into Zoom office hours.